Okay, I can hear that it's recording now. So I'd also like to welcome you to this webinar. This is the second webinar in the series, and we have started this monthly series of webinars that we intend to keep going. And this one will be focusing on visualization. So specifically visualizing 3D spatial patterns using molecular pixelation data. Yeah, and the discussion, how I've structured this whole webinar and discussion is going to be looking at why we do visualization in general, and then also discuss what MPX is bringing to the table in terms of visualization and how it's solving current problems in visualization. So if we start over with why, um, there are currently a lot of limitations in the 2D methods that are applied to visualizing single cell data, and it is 3D visualization is very important in this aspect because things in biology happen in 3D. So we'll go over these limitations and the overall importance of 3D imaging. And then again, we'll discuss a little bit about the molecular pixelation technology, just for those of you who maybe have missed the previous webinar. Then we'll also talk about the advantages with the technology in terms of visualization. And then we'll end up with some practical examples of 3D visualization with MPX data. We'll discuss some of the core principles that guide visualization in MPX data. And we'll look more in detail into one case study that looks in PHA stimulated T cells. So to start with the why and the current limitations in 2D approaches. So microscopy being the dominating approach for imaging and visualizing single cell data has been around for centuries, and it is a 2D approach, and with that come many limitations. Some of those, and this is not an exhaustive list, but some of the big ones here being that it has very low multiplexing, so the number of markers that you can look at one time is very low, and then also very low throughput, especially if you try to increase the plex number. And of course, there's of course efforts in that direction, but most of the microscopy that is being done is not three-dimensional, so there is no z-axis information coming from that modality. Many experiments are still, I mean, there are done in a mono layer of cells. But even if we sort of disregard that, uh, cells are three-dimensional structures. They operate in 3D, so they interact and communicate with other cells, and all of this happens in a three-dimensional space. Some examples I've included here, schematics of how T cells, for example, communicate and interact with antigen-presenting cells when they're recognizing novel antigens for them to mount an immune response. Or another example would be T cells attaching to surfaces, migrating, recognizing tumor microenvironments. All of these things occur in 3D space. So it is instrumental that we're able to resolve this complexity in its native state and look at things that happen in 3D space also using a three-dimensional modality. And this is one aspect where MPX data is it basically shines because it is natively three-dimensional data, and with the protein colocalization and protein polarization metrics that MPX data provides, exploring these three-dimensional events is much simpler. So with that, we'll discuss briefly what the MPX technology can give us, and what it gives us to help us go beyond this two-dimensional barrier and go into the three-dimensional space. So this is just a very quick refresher of how the technology works, uh, just so that we understand better how the data is produced and what it looks like. So the assay is an antibody-based assay using barcoded antibodies, and then these antibodies bind to respective targets on the cell surface. It is an 80-plex assay, so we recognize 80 different proteins on the cell surface. And once the antibody bind to their target, we perform 2 rounds of what we call pixelation. So this is the major component of the assay, is the DNA pixel, which is a rolling circle amplification product. And each of these pixels recognizes 5 to 10 different antibodies, essentially grouping them into these small neighborhoods once the ligation between once the binding between the antibody and the RCP happens. So we basically, in that step, cover the cell in numerous thousands of different small neighborhoods. And we basically perform this step again, but this time we're organizing these small communities into bigger ones. And in that process, essentially, we're mapping out the whole surface of the cell by associating each different protein to different communities and giving it coordinates. In all these 3 steps, we're building an amplicon that is being sequenced, and that is where the data comes from. So after sequencing, we are essentially going from just proteins to these individual small neighborhoods that are then connected into an overall graph structure representing the whole surface of each individual cell. And that is the native format of the MPX data, is this three-dimensional graph that represents the surface of each cell. So the data analysis itself is also quite simple. We have dedicated tools. We have open source software that users can harness to basically analyze the data. And after the software basically runs and analyzes the raw data, you get outputs for downstream analysis, you get a QC report for samples, but most importantly, the data that you get is, again, this graph structure that has 3 different layers of data. So it has the abundance data for each antibody, each marker on the cell surface. It has 2 different spatial scores, so the polarity metric and the colocalization metric. But importantly, it also encapsulates the data that we can use to visualize. And that is the main topic for today, how we can use this data to visualize. So the analysis is basically can be divided into 5 different steps. We're not going to discuss these today because the focus is going to be on visualization. But after we perform these basic analysis steps, then we usually delve into visualization as a last step to completing the understanding of our data. And down here, I have just shown an example of what a annotated MPX data set looks like. These are PBMCs. So in this data set, you can see all the expected lineages, cell types in a PBMC sample. You have CD8 T cells, CD4 T cells, B cells, in all the other cell types that you expect to. But it is important to know that each individual cell on this UMAP embedding here uh, can be a can be visualized in very great detail. So just an example, I've included a CD4 cell and a CD8 cell, and colored you see the respectively the CD4 and the CD8 receptors on those two cells. The pattern here is random, so there is no particular clustering polarization happening on these cells, but you can readily sort of discern what is happening on these cells. So then we go into how we use the MPX data to visualize. And before we go into the actual visualization process, that there is a few things that we need to kind of explain and understand well, a few key concepts, starting with the polarity score. This is one of the spatial scores, the other one being the colocalization score. But with polarity, we're basically trying to see whether there is any clustering pattern of a particular protein on the cell surface. The polarity score looks at a single protein at a time, and it uses the Moran's I statistic to assess global clustering of a protein. It is important to understand that this is a global score, so you basically get a single score, single metric per cell and protein. And we express this metric as a z-score, as we do for the colocalization score as well. And moving to the colocalization score, in contrast to the polarity score, now we're looking at a pair of proteins, so two different proteins, and the colocalization score is basically going to indicate for us whether we see a separation or a segregation between two different proteins, and that is going to correspond to a negative colocalization score. A score of 0 will basically tell us that there is no particular pattern present in on the cell surface, so it's a random distribution of two particular markers. And then a positive score would, of course, tell us that these two proteins are in their vicinity, so they're very close to each other. Um, the colocalization score is also a global score, and it is also expressed in a z-score. And the important thing about why we use that score is to basically make these metrics comparable. There are a few different important aspects that they bring to MPX data. And z-scores in general, they can tell us if a observed spatial pattern basically is different from a what we expect from randomness. But they make our life much easier when we work with this data, because they standardize the data, making comparisons between different cells and different data feasible. And this is not the case when you use statistics such as Moran's I. So having the z-score representation is very important to be able to compare between different samples. Another thing that they bring is robustness. They help us bring down the influence of noise and outliers within the data. And also very important is they help us interpret the data. Z-scores are very standard. So when it comes to hypothesis testing, we know that, for example, a z-score of 1.96 corresponds to a certain significance level. So when we have these scores, we can easily interpret our results from hypothesis testing and from statistical comparisons. So for the rest of the talk, we'll focus on this case study, and I hope with that we'll be able to illustrate the visualization capabilities of MPX data. This is a study, an experiment that we have conducted, and it's been summarized and documented as an application note that you can access on our website. So if you, after this talk, feel that you want to learn even more, you can read the whole application note. But the experiment that was conducted is a comparison between PBMCs that have been either stimulated with PHA or just cultured in a non-stimulated steady state. And with PHA activation or with activation in general, and PHA is a very strong stimulus. With activation, there are certain events that get triggered within cells that basically change their phenotype, their profile. So there are many proteins that need to become more abundant so that the cell can meet its shift and its change of metabolism, but also, and this is not as frequently looked at, is there are many different things, many different rearrangements on the cell surface that do not have to do with changing the abundance of certain proteins per se. And these are the things that we're going to try to look at now with MPX data. So since the focus here is going to be on visualization, we're going to skip a significant portion of the analysis, and we're looking at already an annotated data set. So all of the prerequisites, the pre-processing annotation, these things we're going to disregard for the time being, and we're going to focus particularly on the CD8 T cell subset from PBMCs. And basically, the analysis would traditionally start with doing a differential comparison between the conditions within our experiment, here being activated and resting T cells. But for the purpose of this talk, we don't have to go into that much detail. We can basically just visualize the distribution of polarity score. We'll start with polarity scores, and we instantly see quite a bit of difference. There's a lot happening in activated cells that is different from what we see in the resting cells. For example, here we see on the far right, there's a protein CD82 that has become significantly and markedly polarized in activated cells, but has not changed in the resting cells. And there are multiple other examples of this pattern of polarization that is going on in the activated cells. But one thing that is concerning looking at this data is that we see a lot of markers that are not necessarily associated with T cells. For example, CD20 is a B cell marker, and CD16 is a myeloid marker. So obviously, we need to be aware that using the full 80-plex of markers here is not always pertinent, and we need to do some cleaning here. And these markers that are not necessarily matching the lineage of the cell type of interest are mostly there because they have very low abundance. And we see that in MPX data oftentimes very low abundant markers, very low abundant proteins, they will introduce a lot of noise, which needs to be dealt with. But fortunately, we have the right tools for this. So on the panel, we have four different isotype controls, and those are there just for that reason. We can use those to basically build a reference noise level and filter out anything that is beyond this threshold of noise. So what you see here is just this being done. On the left, you can see the histogram of the cumulative um, expression of the four different isotypes, and then using that, we can establish a threshold here. And when we set this threshold, we see that both CD20 and CD16 are falling to the left of this threshold, so to the side of the isotype controls. And other markers, I've included examples such as CD47, CD2, CD43. All of these are proteins that are expressed and present on T cells. They're basically passing this threshold level of noise. So with those eliminated, now we can focus on actual markers that are relevant for this experiment. And if we look at the top 20 markers here that I've included, we see that now that we filtered the data, we no longer have these irrelevant B cell and myeloid markers present. But we can focus on just a few markers since we can't explore all of them in this in such a short duration. So an interesting observation is again that CD82 is very different between the resting and deactivated cells. But also we have markers such as CD8, and we're looking at CD8 T cells. So looking at and exploring CD8 in more detail makes sense. But that particular receptor seems like it was not affected by the PHA stimulation in a significant way. So let's explore these in a little bit more detail. And here I've basically made a scatter plot where on the x-axis we see the polarity of CD8, of the CD8 receptor, and on the y-axis we're looking at the polarity scores of CD82. And instantly we see that the distribution is very different between these two conditions. So on the side we have the density of each of cells within each group. And if we look at CD8, we see that there's a huge overlap, so there isn't much of a difference in the polarity of that protein. But if we look on the y-axis here, we see that the resting cells are mostly clustered here around 0 in terms of CD8 polarization. But the stimulated cells, those are cover a whole range, but they a lot of them are in the upper side of the range here, so they're very polarized. And as I pointed out early, the nice thing about MPX data is that any single point in this scatter plot represents a single cell that can be inspected in very great detail. So I've included examples of the cell graph layout here for two different cells, and I've tried to basically be consistent in selecting cells that have a similar CD8 polarity score. But you can see that with this activated group, we can go and explore a very polarized example for CD82, but not CD8. And what we see on the right here is the representation of these cells. So if we look at the cell from the activated group, we can see that CD8, the CD8 protein is very much polarized to and clustered in one end of the cell here. If we change and just color the nodes in this cell graph based on the abundance of CD8, then we see that this marker is distributed uniformly on the cell surface. So there's no polarization pattern. And then we're switching to the resting cell, where we see both CD8 and CD82 not showing any distinct polarization. So the data being three-dimensional opens up the kind of the possibilities of looking at these cell graphs from every side and every angle. These are three-dimensional structures. We can do with them, and we can transform them and spin them around any way we want. So this is something that you can easily do with MPX data. And this particular finding about CD8 and CD82, you can read more about in the application note, but we've validated. We're a new technology, so we try to validate all the significant findings that we get that we see in data. And it was what we've documented in the application note. It's expanding a little bit on what I show you here, because we also see that, for example, for CD82, we see no difference for that proteins in abundance. So stimulating with PHA does not induce any change in abundance. However, the change in polarity you already saw, but we also validated that with microscopy. So at the bottom here, you see resting cells and you see the reference. We've used CD8 as a reference here, as I showed you, with the visualized MPX data. So CD8 is uniformly distributed, and so is CD82 in resting cells. But once they're stimulated with PHA, CD82 does not change. However, we start noticing these clear CD82 microclusters on the cell surface. So that's for polarity, but there's more to basically discover with MPX data, and colocalization is also covering a huge part of the data. So it holds a lot of additional insights about the data. And from this particular experiment, we were able to both verify a lot of findings that have already been documented and confirm these, such as the example of CD55 and CD48, which are lipid raft associated proteins, and they have been documented in the literature as being colocalized after activation. So looking at the polarity score in this heat map is showing you polarity score where the pink hue is basically a high polarity score, meaning colocalization. Sorry, high colocalization score, and then the blue is a low colocalization score, meaning a segregation of these proteins. So if you look a little bit closer here and inspect the CD48 and CD55 square in this heat map, you see that there is an increased colocalization score, meaning that these proteins are very close together, and that we're able to confirm with the reports in the literature. We also, similar to the polarization findings, validated this as well using this time super resolution microscopy. And in the example here in the microscopy images, you can see that CD55 and CD48 are basically present in the same sort of spots and clusters here on the cell surface. However, if we look at a different marker, for example, we've used CD7 in this case, it is again being distributed in a more uniform way and not colocalizing with these two markers. Other known things that we were able to confirm with MPX data were established protein complexes such as LFA1, which is composed of CD11A and CD18. Um, you can inspect the heat map and see that there is very high colocalization scores for CD11A and CD18 in the data that is showing the activated cells. Another one is a very well known one is MHC1, that is HLA, ABC, and beta-2 microglobulin. And we also see a very elevated and high colocalization score for that. But it gets more interesting than that because those are just two protein complexes. What MPX data enables us to do is to look at these multi-protein domains, which is very intriguing. And in this case, I've chosen to just show here a few, and those are well-known tetraspanin enriched microdomains, and they're basically parts of the cell surface that are enriched in these proteins called tetraspanins that are very that are instrumental for cell migration and cell signaling in general. So we can visualize the abundance of these proteins on the cell surface, and when we do that, we see that they also colocalize in the same spots on the cell surface. So they're forming these microdomains in this case of three different proteins that I'm showing you here. And we can also look at the colocalization scores that I've included on top just for reference, which seem to be very elevated in for these proteins in PHA stimulated cells. And again, those structures being three-dimensional, we can look at them from all angles, so we can spin them around and inspect closely what is happening and where they exactly being clustered and focused on the cell surface. So this has been just a very brief example of what you can do, but I would urge you, if you're interested in finding out more about how to do this and how to kind of dissect and visualize MPX data in the way that I've shown you, to go to software.pixeljet.com, where we have all the necessary information to get started, even if you don't have data on your own. You can read about how to analyze the data, how to understand the different concepts a little bit more that have to do with visualization. And importantly, we have data sets that you can just try on and visualize any kind of protein or aspect of the data that you're interested in. This particular experiment that I shared with you is also present in the data sets, but there are also other applications that you can explore. Also, we have started a blog that we update quite often, and there you will find little bits and pieces of analysis such as this talk. So there is a blog post on visualization of data where you can find all the code that you need to basically construct these visualizations that I showed you. And with that, I think I have covered what I wanted to, and I would like to see if there are any questions.
Um, I can't see any questions. Maybe we give them some time to write. I can ask a question. So, Stefan, can we visualize cell-cell interactions?
Yeah, you can actually do that with MPX data. There is one caveat in that question, and the answer to it is that you can you can see cells, different cells being attached to one another, but you cannot get into the interface. So if you're interested, for example, into the what proteins are being clustered or kind of focused and centered around this interaction, you can see that for the periphery, but you cannot get into the actual interface. But we have been able to look at different cell-to-cell interactions, for example, between antigen-presenting cells and T cells, or between B cells and T cells. We are able to dissect those and look at those, yes.
Right. All right. Thank you. And I think we don't have any more questions. So we would probably end the meeting here now.
With
Thank you
that,
so much.
I would like to, I guess, add just one more thing, and that is a little bit of a plug and a promotion that, as I mentioned, we have these webinars are a part of a series, so we're aiming to do a monthly kind of release of the analysis webinar. So stay focused, stay tuned, and we'll cover more kind of different aspects, interesting aspects of the MPX data and analysis in the future.
Yes, indeed. Thank you, everyone, for joining, and see you in the next meeting next month. Thank you.
Thank
Goodbye.
you.